Hi, everyone. This is Julie Wolf of the American Society for Microbiology, here with another Microbial Minutes. It's February 5th, 2018, and it's been a full month since our last Microbial Minutes. We've got a lot to talk about. There's been a lot of microbiology-related studies in the news and released in different journals, so let's dive in. What are we going to talk about today? We're going to start with some antiphage systems from bacteria, as well as some antibody information about the Cas9 system. And then we're going to move into bacteriophage that have been found in urine. We'll then talk about some a new index to grade pharma and its abilities to look for antimicrobial, for new antibiotics, as well as a pill that can track digestive gases, or as the title says, a fart tracking pill. We'll move into human fleas as a vector for disease and finish with a new enterococcus virulence factor and what that might mean. So let's begin. Let me just reiterate before we start going through these different reports that you should feel free to add a comment if you want to participate in this live session. Use the chat box. Happy to have this be a back and forth discussion. And if you aren't taking place in the live session, leave a comment and we'll get back to you later. All right, so the first report is from Science, titled Systematic Discovery of Antiphage Defense Systems in the Microbial Pangenome. Take-home message from this report is that new bacterial immune genes will provide biotech possibilities. So I have pictured here a few bacteria that are being infected by phage. Bacteriophage are viruses that will infect a bacterium. And very similar to our cells, they'll use the bacteria machinery, the bacterial transcription and translation machinery, in order to make new viruses. And the bacteriophage looks just like these lollipops here, where the phage genomic material is in that big circle part, and it goes through that long spindly stick into the bacterial cell. And bacteria do not want to be infected by these phage for a number of reasons. We'll talk about the phage lifestyle in a few reports. And they have a couple different defense strategies, or what we refer to almost as immune systems, to help prevent bacteriophage infection. Some of these are really well characterized. You can talk about restriction enzymes or the CRISPR-Cas9 system that you've heard about. Those both cut specific DNA sequences. There are other types of systems, such as the abortive infection system, and that allows a cell to undergo cell death when it senses that it's infected. This is kind of analogous to autophagy, where one cell will die in order to prevent the infection of other cells. So it will save the neighbors. There are other types of systems that have been discovered and are still being characterized, but basically all of these help the cell to recognize an invading piece of DNA and somehow either destroy that DNA or keep that DNA from replicating. And while these are really important in just our basic understanding of microbiology and how biological systems work, these also have played a huge role in biotech. So restriction enzymes have been the bread and butter for how to cut specific DNA sequences for decades, and they're now being usurped or at least complemented by the CRISPR-Cas9 system, which allows scientists to cut DNA not only of bacteria, but all types of different organisms. And so this group wanted to look into other types of bacterial genomes to see whether there were any immune systems or defense strategies that hadn't yet been discovered that might play a role in biotech, or just in better to understand the world around us, of course, as well. So they use bioinformatics to look through 45,000 different microbial genomes, huge number of genomes. So great example of taking all of the data that's out there and sifting through it to look for your research question of interest. And they flagged groups of genes that had unknown function that were located near genes that they already knew were related to defense. And this is because a lot of these systems are quite complicated and require several different bacterial genes that are often kept near to each other within the genome. So a bacteria expression system might have something called an operon, where several genes are activated at once. And so they took these different gene groups and put them into either E. coli or Bacillus subtilis, that's a gram-negative or a gram-positive bacterium, and exposed those clones to the representative phages. And they took different phages from certain classes. Most of them had tails, just like that micrograph that I showed you at the beginning, they had one that was an untailed phage to basically represent the different types of bacteriophages that have been discovered. And from this, they were able to find 9 genetic systems that protect against phage infection, and one system that protects against plasmids, which is uh, potentially interesting in the biotech world because people use plasmids in order to move and manipulate DNA in almost every single lab that does molecular biology. And so this might have some sort of implication with transformation efficiency or something along those lines. They didn't really characterize the mechanism of all of these different systems because, of course, they found so many of them, they couldn't follow up on each of those systems. But they did look for regions of homology so that they might be able to see what some of the mechanisms are of these different systems. And here, the one of the systems that they characterized is called the Zoria system. This was covered by several outlets, so I wanted to show an image. And the proteins, when they're expressed, have a similar shape to proteins that are involved in bacterial flagella system. So a flagella is a long, kind of wavy appendage that bacteria will use in order to propel themselves through a liquid medium. And these particular genes are not involved in moving bacteria around, but what the researchers suspect is that when a bacteriophage is invading and detected by the bacterial cell, that the bacteria can depolarize its membrane. So there's this electrochemical gradient across the membrane, which is necessary in order to make ATP and make energy for the cell. And by stopping that process, the cell will basically die. And so this is one of those potentially cell death mechanisms that would prevent the phage from making more phage that could infect neighboring cells. And so this vastly expands the number of known mechanisms of bacterial immunity and was shared both by the lab, which did the work, as well as others that were very excited to see this work done. It was picked up by several trade outlets. And here there was another researcher who was not involved in the studies who was excited to say that this has more than doubled the systems that we know that are involved in phage defense. It's really quite a feat of bioinformatics. So we're going to move on to another um, bio bacteriophage defense system and talk about CRISPR-Cas9. Now, this is from a paper that has not been peer reviewed. This was posted on bioRxiv, and it's titled Identification of Pre-Existing Adaptive Immunity to Cas9 Proteins in Humans. The take-home message is that human antibodies to Cas9 may impede CRISPR therapies. And the reason I'm including this, even though it's not yet peer reviewed, is because it really brings up quite an important message. And this take-home message has already been covered by several outlets and undergone conversation on different social media platforms. So I thought it would be worthwhile to highlight. Now, a lot of those defense systems involve several bacterial genes and are quite complicated. So if we see on the left here, this is what happens when a phage, as I showed you before, injects its genomic material, often DNA. And there are a number of different proteins and pieces of RNA that are required in order to basically chop up that phage genome material. Now, researchers have been able to simplify this greatly by using the CRISPR-Cas9 system. And so this requires basically 2 components. Here we see double-stranded DNA. This is what the researcher wants to cut. And you need to include, in order to cut this, a protein called Cas9. That's this kind of tannish blob in the back. That's the endonuclease that will actually nick the DNA and allow the 2 different pieces to be separated. And you also need a guide RNA, which is kind of an assemblage of different RNAs that are used within this more complicated system. And basically, the guide RNA programs that Cas9 and gives it the target sequence that then the Cas9 protein can find and cleave. Now, if we're going to use this in any type of genomic, any type of clinical study where we want to change the genome of people, there could be a real problem if there are Cas9 antibodies that might bind to this Cas9 before it gets into cells, or even potentially lead to some sort of autoimmune response and big inflammation that could potentially be damaging to the nearby tissue. And so these researchers wanted to ask whether or not those antibodies, and they also looked at T cell responses, whether those exist in people already. And in fact, they do. So they took a number of different patient sera, just looking at antibodies that are circulating in the blood, and they saw that there are antibodies that exist in a number of people to Cas9 enzymes from two of the most common organisms that are used in these genetic studies, Staphylococcus aureus and Streptococcus pyogenes. And it kind of makes sense that we would have antibodies to this protein already, because both of these organisms, although they can cause disease, these bacteria can also just survive on our skin and our oral cavity as commensal organisms that are not causing disease. And then we would be, the people who are colonized with them would be exposed to the different antigens that are associated with these bacteria. So they saw, in general, seropositive or a positive antibody response from 79 people who were infected with, who had antibodies against the Staph aureus Cas9, and 65% of people who had antibodies against S. pyogenes Cas9. So what does this mean? Does this mean that we are not going to be able to use CRISPR-Cas9 in our genetic manipulation studies? Probably not anything quite so severe. So this, as I mentioned, was covered in several different outlets. One of the lead authors was Matthew Porteous. He was interviewed by Andrew Joseph for Stat magazine, and he said here that, like any new technology, you want to identify potential problems and engineer solutions for them. And I think that's where we're at. And in another outlet, Porteous goes on to say, one potential solution is to develop a Cas9 system from bacteria that do not colonize or infect humans. And that's true. That's one way that we could get around this system and avoid having antibodies that will impede the genome manipulation process. There are other things that we could imagine, such as site-directed mutagenesis to change the antigenicity, or the way that the Cas9 is recognized by our bodies. And fortunately, a lot of the clinical trials that are approaching the beginning of their trial do not actually involve injecting Cas9 into people. A lot of them involve taking cells from people, cells such as bone marrow cells or white blood cells, manipulating them with CRISPR-Cas9, and then putting those altered cells back into the patient. So the Cas9 would no longer be in the system and hopefully would not be even exposed to people in the first place. Now, interestingly, as soon as this bioRxiv non-peer-reviewed article was posted, it was noted that company stock of companies that involve CRISPR technologies was already decreasing. We'll see how this plays out in the future. So we've talked about bacteriophage defense systems and how those can be used as tools. But bacteriophages themselves can also be used as tools. And that was highlighted in a recent Journal of Bacteriology paper, which is an ASM journal, titled Bacteriophages of the Urinary Microbiome. The take-home message here is pretty broad. It's that bacteriophage abound within the female urinary tract. And this is a very understudied area in looking for bacteriophage, which is why this is an important paper. So bacteriophage are known to help shape the microbial community. And that's because, as I discussed earlier, they can infect a cell, such as we see here. There's a bacteriophage injecting its genomic material, which will then incorporate into the bacterial genome. And because the bacteria cannot differentiate the phage DNA from its own DNA, it will transcribe and translate the different RNAs and proteins necessary to build new bacteriophage and assemble those. And eventually they'll accumulate to such a large concentration that they will lyse or break open that cell and thus kill that bacterial cell. But sometimes the bacteriophage can incorporate their DNA into a bacterial cell, and it can just quiescently exist within that genome without making new progeny phage. And in this case, it allows that phage DNA to be replicated every time the bacteria DNA is replicated. And every time the bacterial cell divides, each daughter cell will receive that, what is now called a prophage or incorporated phage DNA with the bacterial genome. And so you can imagine, after several generations, if the bacteria is replicating many, many times, and of course, bacteria replicate very quickly, between 20 minutes and maybe 2 hours, depending on what type of bacteria you're looking at within the gut. You could imagine that a large proportion of the bacteria would have this prophage within their genome, their genomic sequences. And sometimes these bacteria with what is called lysogenic bacteriophage can be stimulated to activate that prophage and start to express the viral proteins and generate that viral genomic material. And you could imagine that if a large population that has this prophage in their genome is activated to undergo this lytic cycle, a bunch of bacteria are going to die, and this can shift the microbiome community. And so this has been shown to play a potential role in disease in the gut. And so these researchers wanted to look at the potential role of phage within the urinary tract, where, especially in females, urinary tract infections are a big source of infection. So what they did was to take 181 isolates from different bacteria that grow in the female urinary tract and to look at their sequences and investigate for what would appear to be prophage DNA. And I'm showing you here at the bottom some of the phage that they were actually able to identify and take a picture of using an electron micrograph. This is the phylogenetic tree, not of the phage that they identified, but of all the different bacterial types that they looked at. So you can see here's the firmicutes over here, the proteobacteria at the bottom, and the actinobacteria over here on the left. And within this tree, every time you see a line, that's a different bacterial strain. And if there's a circle on that line, that's indicative of a prophage that was found within the genome. So you can see that majority of these had at least one circle, at least one prophage DNA, and some had more than one. And the different colors indicate whether that prophage DNA was identified in the bacterial genome itself, or whether it was incorporated into a plasmid DNA, and it also has to do with how certain they are that that is an accurate prophage. And so what they found were 86% of these isolates that had at least one and sometimes more phages associated with their genome sequence. And so this was shared by different outlets and on social media. Ars Technica covered it. They do a great job of incorporating puns into their titles, saying that you're in for an interesting story. Never before seen viruses that can kill bacteria stream from women's bladders. And here the lead author, Putanti, says that she hopes to eventually characterize these a little bit better and then ask whether you can use these phages to shape microbial communities. And implied here is that we would be able to shape microbial communities into something that was beneficial for health. The author of this piece, Beth Mole, also made a lovely Parks and Rec reference. Leslie Knope was famous for calling the bathroom the Whiz Palace, and here she says that the Whiz Palace overflows with viral gold, apparently. This was also shared by other phage researchers, such as Stephanie Strathy, who has a very interesting story about the use of bacteriophage. I recommend you find her TED Talk if you haven't. All right, moving on and away from bacteriophage, we're going to very briefly cover a press release that was titled Superbugs, First Independent Comparison of Pharma Companies' Efforts to Address Drug-Resistant Infections. Take-home message from this is that a new index will grade pharma on their efforts to develop antibiotics. And this is the first independent analysis of the pharma industry and their efforts to tackle drug resistance, which is a huge problem. We talk about it all the time here on Microbial Minutes. Almost every microbiologist knows that antimicrobial resistance is increasing in the clinic, particularly with bacteria. And so this group, which was funded by, I believe, the British and Dutch governments, is going to publish, and they published the first antimicrobial resistance benchmark, also called the benchmark, which was published by a nonprofit group that it's called Access to Medicine Foundation. And they also make another index that grades different pharma companies on their ability to provide medicines to low resource countries. So their key findings were pretty straightforward. They found that there are 28 antibiotics in later stages of development. And these antibiotics target pathogens that are deemed critical antimicrobial resistant targets by the WHO, the World Health Organization. We talked about that report that came out on a previous Microbial Minutes. They found that Pfizer is running the most drug development programs. And then a few other things that are a little more related to the direct development of antimicrobial resistance. They found that 8 companies are setting limits on the concentration of antibiotics that manufacturing wastewaters, so that's the wastewater from plants where they're making these antimicrobial drugs, And there's a limit that is set for how much of that drug can be put in the wastewater before it's released into the environment, which is really important because if we're dumping a lot of drugs into the wastewater, that will eventually flow into other water systems and continue to select for resistant isolates. The benchmark also found that there are 4 companies taking steps to separate the bonus of a sale agent from how many or how much antibiotics they sell. So, of course, with sales, generally you are rewarded for selling more of a thing, and that might perhaps induce some individuals to offer antibiotics when those are not necessarily needed. And so by separating the pay and bonuses from actually how many or the volume of antibiotics sold, this is hoping to better steward exactly using antibiotics when they're needed. This was covered by a number of different outlets. Of all the stories that we're talking about, this is probably the most covered by mainstream news, such as the New York Times. And it was also shared by different Twitter and social media users who follow antimicrobial resistance. All right, so I promised you a fart tracking pill, and here is the study itself. It was published in Nature Electronics, and it's titled "A Human Pilot Trial of Ingestible Electronic Capsules Capable of Sensing Different Gases in the Gut." So this new pill tracks digestive activity. Take home message: that digestive activity, of course, can be excretions from our own digestive system, but a lot of it comes from the microbial activity of different microbes that are helping to break down the complex molecules into things that are more readily absorbed by our gut, and the byproduct being these different gases. Now, this pill is super cool. You can see how big it is here in the lower right hand side. There's this hand that's holding it. So it's like a very, very large fish oil pill. This is going to have a little bit of difficulty in swallowing, but it is ingestible. And it has many cool components. The actual shell of the pill is a non-permeable polymer. The only place where gas is read is at the little filter at the front here, where different gas molecules can be read by the gas sensor. And these can then be transmitted. So, what the sensor is detecting can be transmitted wirelessly. So, you can control this and get the readings. Let's say, for example, if this is incorporated into some sort of iPhone app, it'd be pretty cool if you're able to track that just in order to look at your own gut health. So, as a proof of principle, they gave this to an individual and followed it as it passed through their gastrointestinal tract. So, this is not something that sticks with you. You're basically going to swallow it. It will go through the long tube of your gastrointestinal tracts, stomach, duodenum, small and large intestine, and then eventually be excreted, right? And so the here on the left-hand side, I'm showing the first individual who was brave enough to ingest this pill and have their digestive tract followed, and you can see that they were able to detect oxygen, hydrogen, carbon dioxide, and temperature. So these top 3 are more indicative of metabolic activity, but temperature is also quite interesting. One of the things you can see right away is that as the pill is passing through from the stomach to the different parts of the intestine, is that oxygen levels drop off precipitously into the colon. That large intestine is really an anaerobic environment where there's not oxygen. And in fact, the microbes that live in that environment want there to be they work best. And then some of them even require that there is no oxygen present in order to do their metabolic activities. So you can see that very well with this gas sensor. The researchers also denoted when certain events happened with this individual. So here's where lunch happened. Individual had a snack. Here's dinner. Here's where he or she went to sleep. And also, interestingly, the temperature fluctuated just a little bit. So 37 degrees Celsius is body temperature. But of course, this is a long tube we're talking about from the from the intake to the outtake. And you would imagine that those areas that are not surrounded by our warm guts and our innards would be a little bit cooler. And in fact, they are. You can see toward the entrance and toward the exit of this pill that the temperature drops just a little bit, while in the depths of our bowels, it is nice and toasty. They were then able to use this pill to look at some experimental data looking at reproducibility in a few different volunteers and even giving the volunteers different diets. So here we have 2 volunteers who were given a low fiber diet and 2 volunteers that were given a high fiber diet. And you can see right away that here we can see where the pill was excreted after going through the colon, right? And it has that same pattern where the oxygen drops at the end in the colon. Here, although the time goes roughly 24 hours, it actually takes these low fiber diet individuals much, much longer to excrete this pill than it does the high fiber diet individuals. These two excrete the pill within around 20 hours, which is what we see over here. And it took the low fiber diet individuals about 3 days to excrete that pill. So not only is fiber, which we've talked about many times on Microbial Minutes, not only is fiber important for gut health because it helps to select for a diverse microbiota, but it also seems to select for gut activity, which I think we all know. This is something that's pretty well known. High fiber diet helps keep things moving, but it's another data point to support this type of activity. And you can only imagine that going forward, you would be able to use this in order to test many different types of diets and lifestyles and how that can affect microbial activity in the gut. So this was covered by a number of places. There are science journalists who are promoting this on Twitter. This was covered in a YouTube video. It's about 6 minutes long called Nerd Alert. I don't have a link down below, but it's pretty easy to find. And it was also covered by a commentary in the same journal, Nature Electronics, where the writer of the commentary, the author, Benjamin Terry, had a very interesting idea for the future of going to get your annual physical, where you might, in addition to getting traditional readings like blood pressure and temperature and having a stethoscope to measure your heart rate, you might also be given this pill maybe beforehand or maybe during the excuse me, during the visit. And the doctor would then, or the healthcare provider would be able to look at the readout in order to see your gut gas profile and how healthy your microbiome is, maybe making suggestions for your diet based on that data, which is quite an interesting idea. We'll see how accessible these things become going forward. Excuse me. All right, moving on to the next story, we're going to move into a PNAS paper that was titled Human Ectoparasites and the Spread of Plague in Europe During the Second Pandemic. Take-home message of this is that human fleas likely helped transmit medieval plague. Now, I don't know about you, but I did not realize that humans had fleas. That was one of the things that I learned in this research, but let's back up a little bit. Plague is something that we're all familiar with as the Black Death caused by Yersinia pestis, the gram-negative bacterium. It can be in the bubonic form where it needs a vector to transmit it. So that's traditionally been thought to be fleas that travel on rats. It can also be a pneumonic plague, and that's where it becomes transmissible by aerosols that are spread by coughing once the organism is colonizing in the lungs. Bubonic plague has, for a long time, as I mentioned, been thought to be spread by fleas that travel on rats. So rats are part of that infectious cycle. But this research suggests that human ectoparasites, including human fleas and body lice, which one could be maybe a little more familiar with, lots of little kids get lice, are thought to spread it rat to person from person to person directly. And so I have a picture here of a flea. Part of the Yersinia pestis cycle is to infect the flea. So the flea is not just carrying it, but the flea is actually infected. And if I recall my plague biology correctly, I believe it causes blockage of the flea intestine. So when they go to try and take a blood meal, they actually regurgitate, and that encourages some of the Yersinia pestis that they've ingested to then enter into the bloodstream of the person that they're trying to bite. Excuse me. So there have been 3 major pandemics. We covered this a little bit in a previous Microbial Minutes where we talked about the origins of Yersinia pestis. There was the first pandemic, the Justinianic plague. The second pandemic, this is the Black Death, which is associated with killing 1/3 of the population of Europe. And then the third pandemic, which began in the beginning of the 19th century. this second pandemic is a little bit different because of its very high mortality rate. And also, unlike the other pandemics, it was not associated with rat falls. So these are epizootic events where, in addition to a lot of people succumbing to disease, there would be piles of dead rats that had been part of that infectious cycle where the fleas came from the rats, but the rats were also infected and killed by the disease. So these researchers wanted to look through old data to see whether they could determine by infectious disease modeling and mathematical modeling of diseases whether it was the fleas from the humans, fleas from the rats, or maybe even pneumonic plague passed person to person that was responsible for this very high death rate and no rat falls. So they took studies that had been previously done looking at plague mortality rates in the second pandemic from different locations. You can see up here in the table different cities that were studied that had suffered different epidemics that are from these dates. The previous research had found the population of the city at that particular time was a certain number, and they looked at how many people died. The researchers of this study then tested three mathematical models for transmission, looking at that human ectoparasite, the rat flea, or pneumonic, the human to human transmission. And they modeled it, as you can see over here on the right hand side. So, really, what we're comparing here, because the old hypothesis was that it was this rat and flea model, it was the green to the pink, which is the human ectoparasite model, with the lines indicating what they would expect and the actual observed deaths being demarked with these black dots. And you can see it's more obvious in some cities than in others, but Florence is a good example where the dots very clearly align with this human ectoparasite model compared to the green, which would have a much sharper increase that would indicate the rat and flea model. Other cities that fall very clearly into this pattern are Malta. I mean, all of them fit in general into the ectoparasite model more clearly than they do into the rat and flea model. But in my mind, or by my eye, I can more clearly see it in some of these cities than in others. And from this data, the researchers summarized and said that in general, the human ectoparasite model fit the pattern of the observed data for the second pandemic outbreaks. And so this was shared by a number of researchers and a few different outlets. So, another microbiologist who looks into the history of microbiology said that he'd been arguing this for the last 10 years, that Black Death is spread by human lice. And there's a nice behind the paper. So, this is a blog from Nature. And this is where the researchers talk about their own research and what they've been able to find in more lay terms than the technical reports. And Monica Green, who's a medical historian, and I recommend following her if you're interested in this kind of information, talks a little bit about using images from historical documents. And the behind the paper blog post is quite interesting as well. One of the authors, Catherine Dean, says that the results provide quantitative evidence that challenges the assumption that rats spread the plague in Europe during the second pandemic. We hope that additional studies will investigate the potential of human ectoparasites as plague vectors and the roles in past pandemics and help us rewrite history. So this is cool research that has the implication that it may rewrite the history books, but it also has some cool implications for understanding how different epidemics appear and how do they disappear. And that's really important given that plague is a disease that we're still living with. There is an ongoing epidemic that is, or an outbreak that is occurring in Madagascar, and understanding why this disease pops up sometimes and then will disappear again. I will be very useful for people to plan how to treat outbreaks in different areas of the world. All right, we're going to jump into our last story for this Microbial Minutes. This is from Cell Host and Microbe, and it's titled Identification of a Botulinum Neurotoxin-like Toxin in a Commensal Strain of Enterococcus faecium. So, take home message is Enterococcus gains a virulence factor, but there's a little more to the story than that. So, botulinum neurotoxins, you may be familiar with this as Botox, but these are not things that are only used for cosmetics or other medical procedures such as treating migraines. This is actually a very potent toxin, one of the most highly regulated in studying because it can so quickly have an effect on the host, and it's a potential bioterrorism agent. So these botulinum toxins target neurons, And it does that by blocking neurotransmitters. If you see here on the right-hand side, inside of a cell, there are synaptic vesicles. So this is inside of a neuron. And all of those neurotransmitters need to be passed from one cell to the other. And that happens by the fusion of membranes. This transmembrane protein here called VAMP2 needs to help fuse this membrane with the cell membrane to release these different neurotransmitters. And what these botulinum toxins do is to cleave the proteins that are necessary for the fusion and transmission of these neurotransmitters. Most of these botulinum neurotoxins are found in different members of the very diverse genus Clostridium. So Clostridium botulinum is probably the most famous, but other Clostridial species can also have similar or related neurotoxins. But what this report finds is that this botulinum toxin-like toxin is found in a commensal Enterococcus faecium strain. And the reason why this is something that I thought worth highlighting is because Enterococcus, both Enterococcus faecium and other Enterococcus species, are experts at sharing mobile DNA. They take up DNA from the environment and they pass it on to others, and they are very promiscuous. They don't only share it to other Enterococcus species, but they can share across different bacterial species. So that means that this particular commensal organism, although it may not use the Botox-like toxin as a virulence factor in its own infectious disease cycle, it may pass that piece of DNA containing the new virulence factor onto other bacterial species. So let's back up and take a look at the lovely graphic abstracts. I really like these from the Cell family of journals. What this group was doing was not even looking for Botox-like toxins. They study Enterococcus and the evolution of Enterococcus resistance to drugs, as well as its different types of virulence factors. And so in studying Enterococcus that they had isolated from a farm, they looked at the sequence, compared that to those in public databases, and found that there was something that looked very much like a botulinum neurotoxin. Not only did they find it within this isolate, but they found it on a conjugative plasmid. So plasmids are much easier for bacterial species to share than pieces of DNA that are already on their chromosome, that are integrated into their genomes. And so this is yet another worrying point in the Enterococcus having this particular piece of DNA. Not only do they have it, but they have it on a plasmid. They looked at its relatedness to other botulinum neurotoxin-like molecules, and they also looked at the type of bacterial species. So there's different Enterococcus faecium isolates, some strains, and some of those are known to cause disease and often are associated with multi-drug resistant infections. As you see here, this was fortunately a commensal, not often associated with disease. It resides within the gut as one of our commensal organisms. But as I said, these different Enterococcus species can share DNA quite readily. So this commensal organism may pass it on to one of these multi-drug resistant strains, and it may even pass it on to another species. However, although they were able to confirm that this new toxin does cleave the same type of proteins necessary for neurotransmission, it cleaves it in a different place. It has a different cleavage profile, and they didn't show that it works in vivo. So they didn't really put it in. They didn't really put it into people to confirm that it would work on human neurotransmitters. So that's an important point that I think will be followed up in future studies. This was shared by one of the authors who said that an active botulinum neurotoxin in this commensal, sometimes in its escape, a group of bacteria that are highly associated with multi-drug resistant infections. And this researcher also pointed out that the last time that something worrisome showed up unexpectedly in Enterococcus, which was vancomycin resistance, the Enterococcus transferred that resistance to Staph aureus. So, of course, we hope for a different outcome, but it's important to track now that we know that Enterococcus has this piece of DNA. This is highlighted in a few different outlets. Here I have some quotes from a different researcher involved with this study, who says that Enterococcus is a central hub for gene transfer within the gut, and that makes it potentially scary, as I mentioned. And the lead researcher, Le Breton, here says that it's possible that this particular toxin or even other novel toxins may have been there all along. This will turn up in other enterococci isolated from the wild. We just never looked for these before. So before we totally panic and worry about Botox being contained within this enterococcus, let's make sure that this isn't something that maybe serves a different purpose or does not have an effect on our cells or may have been there all along in different isolates. So that's going to wrap up what we've learned today in our microbial minutes. To summarize, we learned that bacterial immune genes will provide biotech possibilities, that human antibodies to Cas9 may impede new CRISPR therapies, but we'll see about that. We heard about bacteriophage that abound within the female urinary tract. We heard that there's a new index that grades pharma on their efforts to develop antibiotics. And we heard about this nude pill that tracks digestive activity. We learned that human fleas likely help transmit medieval plague and that Enterococcus has gained a virulence factor. I'd like to thank you for listening. If you have any suggestions for papers to cover in future microbial minutes, please do suggest those. Leave a comment on something that you find interesting. We're going to try and do this in another 2 weeks because there's just been so much really cool microbial science coming out. So I'd like to do another one of these in February. And until that time, happy for your feedback, and I'll talk to you next time.